The use of tissue-specific expression of polyglutamine fluorescent reporter in C. elegans is significant because it allows the discovery and characterization of proteostasis regulators in the context of an intact multicellular organism.
The main advantage of this technique is that it allows visualization and quantification of the age-associated decline in cellular proteostasis in vivo, which is essential to gain deeper mechanistic insight into how organisms maintain proper folding and function of the proteome and the effects of aging.
Elucidating mechanisms capable of preserving proteostasis will facilitate the development of targeted interventions for the treatment of aging-associated diseases in which proteostasis is compromised and to promote healthy aging.
Proteostasis collapse is a massive clinical problem as it underlies the development of protein misfolding diseases, including Alzheimer's, Parkinson's, Huntington's disease, and amyotrophic lateral sclerosis.
Begin by using 6 cm diameter Petri plates to prepare 5 agar plates for each test condition. For experiments with RNAi, induce dsRNA production in transformed HT115 E. coli and use RNAi agar. Use OP50 E. coli on standard NGM agar for experiments without RNAi. Grow the E. coli cultures overnight at 37 degrees Celsius while shaking at 220 rpm. On the next day, pellet the bacteria by centrifugation at 2400 G for 15 to 20 minutes. Then, aspirate the supernatant and